Recording now, ladies and gentlemen, we're going to review cells and cell structures. And this comic right here is a really funny joke. I don't know if you notice, but it's great. Transport proteins. We're gonna talk about this later. We're gonna talk about this later. Don't you worry. So, oops, how did that get there? Okay. I'm going to briefly go through the organelles. I don't have all of them written out here. I actually, on the next slide, there's a link to a Quizlet where you could review the exact functions, and it's got it all written out and things like that. But I'm going to show you in the cell where everything is and briefly review what everything does. So starting with here, nuclear envelope, it surrounds the nucleus. Nuclear pores are needed so things can get into and out of the nucleus. But in here, that's where DNA is stored, meaning that's where transcription must occur. Also, there is nucleolus in here. These make ribosomes, which will then travel out to the rough endoplasmic reticulum, these little guys here, but also in the cytoplasm as well. They're going to create the proteins, which is kind of the unit that we're on right now. Smooth endoplasmic reticulum, there is lipids that are made here. There's a little bit of detoxifying that occurs in the smooth endoplasmic reticulum as well. Cytoplasm is the space that is not organelles. It's just kind of there's a lot of stuff going on there, but it is the space. I talked about the rough endoplasmic reticulum as well. This is going to be where proteins move through and where proteins are made. Talked about ribosomes, the cell membrane. We're going to talk a lot about today. A lot of FRQ questions are based on the actual transport into and out of cells. Mitochondria, you know about these endosymbiotes, neat little previous prokaryotes give us energy. Centrosomes in animal cells are going to be how cell reproduction occurs. And within a centrosome, the whole structure is one centriole. Microfilaments, sometimes called actin microfilaments, are a type of cytoskeleton. Microvilli are little extensions of a cell to give more surface area. Secretory vesicles are vesicles, basically boxes, or bubbles that contain things. If they're secretory, they're going to help move things out of a cell. Microtubules, they help in cell reproduction as well. They give structure to cells too. Golgi apparatus, this adds things and modifies proteins before they leave the cell or go to other places in the cell. Lysosomes are your garbage disposals. They're going to clean up old stuff. They're going to break things down. There's a lot of them in macrophages, which are immune cells. Peroxisomes, these are detoxifying centers. They're going to definitely break things down with peroxides. But yeah, these are like special vesicles that break things down. And we're back to the nucleolus again. And again, that was a real speed through. But if you want to spend more time with it, I recommend following this link so that you can practice understanding those functions. The most common way that I see really organelles being asked about is kind of in a figure like this, where you have to understand the process by which a protein that is meant to leave the cell starts its function and starts its path of existing. So in the nucleus, there is DNA stored. It gets transcribed into mRNA, which actually gets transported out of the nucleus and gets translated into proteins on ribosomes in the rough ER, or out into free floating ribosomes as well. But a lot of them are in the rough ER. Then after that, it's going to be a part of a vesicle, one of those bubbles that moves to the Golgi apparatus, where it gets changed a little bit. And then after it has been changed a little bit, it gets marked for exiting from the cell or being secreted in another secretory vesicle. That vesicle is then going to move towards the cell membrane, where it's going to fuse with the cell membrane and leave. I got this whole thing from a paper on insulin. So insulin, obviously needs to be secreted from cells. The gene for insulin is in your nucleus. It is, transcribed, translated, it's called minus insulin before it is modified by the Golgi into the full form insulin, where it's then pushed out of the cell into the bloodstream. And this is over here, if you have a protein that's wrong, it gets modified. You don't need to know this word too much, but it gets modified by something, and eventually, if it's wrong, is going to be targeted, and the lysosome is going to break it down. Old mitochondria that don't work anymore could also be broken down in this way. So do I have any questions on that so far? We're going to talk about that more, actually. That's kind of what SMB was at the end, but any questions so far? Okay, not hearing any. I'm going to talk about transport, because this is a really, really important part of understanding cells, is getting things into and out of cells. And that is typically referred to as transport by the cell membrane, which can be active or passive. Now, the cell membrane only allows a certain number of things in. And I really like this figure despite the watermark. Please forgive me. But things that can easily get through are going to be small hydrophobic molecules or nonpolar molecules, such as oxygen, CO2, nitrogen gas. Again, these are small nonpolar molecules that can just get right through there. Small uncharged polar molecules like water or ammonia or glycerol, these are pretty small. They can get through. Oftentimes they need some sort of transport protein. Look, the joke came back, and I'll talk more about it later. But yes, they can get through with the aid of proteins because they're small and they're uncharged. Large uncharged polar molecules like glucose or sucrose can almost never get through all by themselves. They almost always need some sort of channel protein to get through. It's going to be very, very, very rare for that to occur. But again, channel proteins can get them from one side to the other, because you need glucose from outside the cell to get into the cell, so you can do cellular respiration, other things. But ions cannot. Ions are charged. Because they are charged, they cannot get through this hydrophobic center here. Really, really polar. So they are not going to get through it all. If you need to get ions across, you need to use a transport protein. So speaking of transport, again, passive transport. It doesn't require energy. Things move with the concentration gradient or down the concentration gradient. Basically, if you have a lot of something in one area and simple diffusion can occur across the membrane for that molecule, it will. It's going to go from high concentration to low concentration. That's with the concentration gradient. That's just called diffusion. And those small nonpolar molecules like CO2 and oxygen can get through with diffusion just fine. On the flip side of the coin is active transport. That's where you want to build up a lot of something, like in cellular respiration, the electron transport chain, where you use energy to make the protons go from one side to the other side, or something like a sodium-potassium pump, where you want a lot of sodium on one side and a lot of potassium on the other side. If you remember from psych, or if you've taken psych, you need that in order to establish like a resting potential in nerves so that you can fire off signals. But anyway, you use something called pump proteins or protein pumps, like the sodium potassium pump, where it takes ATP to make sodium go from where there's not a lot of it to where there is a lot of it. So you can build up that concentration gradient. But again, it requires energy. And a sodium potassium pump, it's great. We haven't talked about it too much, but it's great. It's going to build up potassium on one side and sodium on the other side. So it's really working great to build up concentration gradients on both sides. That's active transport. So talked about passive, talked about active. Simple diffusion of small hydrophobic things getting through, that's diffusion. Requiring something that is very low to move to where it's very high, that's going to require energy, and that's active. But if you have a lot of something going from one place where it has a high concentration to where it's a low concentration, but it can't get through the membrane, you could still have diffusion, but it has to be something called facilitated diffusion, where you use a transport protein to go from high concentration to low concentration. And it could be a channel protein where it can just get right through, or it can be a carrier protein like this. But again, these are just transport proteins that enable you to move from a high concentration to a low concentration if you can't get through the membrane, like a small or a large polar molecule that needs to get to the other side. Okay. And then, speaking of small polar molecules, there's water. Water is definitely a small polar molecule, but it can actually get through the cell membrane because of aquaporins. They are a channel protein. But water will move. You need water in lots of areas throughout the body. So water will move from where it is high to where it is low. And there's a special name for that called osmosis, where water moves down its concentration gradient from where it is high to where it is low, through, again, those channel proteins, or sorry, I guess they're channel proteins called aquaporins. But that's called osmosis. And we did membrane potential earlier, where we did calculations with that, but I just want to relate it back to the structure of the cell. And the implications of that in cells are, with tonicity, if you have a very high solute area where water wants to move into that area, that can actually cause water to rapidly leave the cell, and you get these cells that look like this kind of beef jerky looking cells, because they've lost all their water. If it's an isotonic environment like saline or Gatorade, then the water is just going to go same amount in, same amount out. That's isotonic. And then hypotonic in animal cells is real bad, because that means if you put your cells into where there's too much water, where it's like pure water, the water is going to rush into the cells and they're going to burst, like in my horrible experiment. But in plant cells, that actually works out okay, because they have this wall that makes sure that they're not going to burst. So actually, this is something that plants employ, that they're going to actually pull water from one cell to the other cell, and water is going to rush in order to make sure that the plant stays turgid and upright. If you've ever seen a wilty plant, it's usually because they're not getting enough water. They want to be in very watery environments in order to help them out. But anywho, that is tonicity, and it's all due to, again, osmosis, water moving from where it's a low concentration, a high concentration to a low concentration. And then something we haven't talked about too much, but something I wanted to bring up here while we're here, and we'll hit it again later in the year, but this seems like a reasonable time to cover it, even if it's in a review, is cell surface area to volume ratios, where the reason cells are small is because the smaller you are, the more easily things can diffuse throughout you. So in this cube here, if we have really big cells, we could pretend these are cells, but they're cubes. Then you see that the middle stays pink. This is actually an experiment where they took these and they had an indicator, and then they saw if the outside could diffuse through. So diffusion reached the little one all the way through, but on the next size up, the middle did not quite get diffused to. The molecules couldn't get there. You go even bigger, the molecules, there's even more volume that doesn't get diffused to, and it's the largest means that there's even less volume that was diffused into, and that's because the smaller you are, the more surface area you have relative to your volume. So there's a lot of space that can be accessed in a small thing relative to this, where the stuff on the very inside doesn't have access to this here. It's kind of like a city that if you have a really big city and you live in the middle of the city, it's going to be harder for you to get outside of the city, get things from outside of the city or to get things out of the city. Whereas if you live in a real small city, it's real easy to get into and out of. And that's kind of how cells work. But again, it's relating the surface area, how much space or, surface area around it there is relative to the space inside of it. And then this also is employed by elephant ears. Elephant ears are full of blood vessels, and they're really big. So that allows, anytime an elephant gets hot, the blood is going to carry that heat into their ears, and it's going to be easily able to diffuse because there's a lot of surface area that it can diffuse across. All that blood can easily come in contact with the air where it can get rid of its heat. Okay, so I think that might be the end of the things I have to say. Do you all have any questions about any of these concepts? Okay. All right. Y'all are rock stars. You ready to get to that FRQ practice? Okay. So we can do that. So I'm going to stop